Assalamualaikum warahmatullahi wabarakatuh and selamat sejahtera to my honorable judges. Today I would like to explain about my invention, which is encapsulation of FICUS DELTOIDEA or COTEK extract with hydroxyapatite nanoparticles for cancer treatment. My name is Associate Professor Dr. Midhat Nabil Ahmad Salimi and my team members are Associate Professor Dr. Muhammad Shabil Ahmad and Wafida Khalid Ahmad. We are from University Malaysia Perlis or UNIMED. So this is going to be my outline for my presentation. I will start with introduction or problem statement, followed by product description, novelty, results, commercial potential, and publications. So I would like to start with the bigger picture of my research or the problem statement, which is cancer. Cancer is a genetic disease that is caused by changes to genes that control the way our cells function, especially how they grow and divide. Cancer is the second leading cause of death globally and is responsible for an estimated 9.6 million deaths in 2018. Globally, about 1 in 6 deaths is due to cancer. The total annual economic cost of cancer in 2014 was estimated at approximately US $1.16 trillion, and this cost will keep increasing from year to year. There are many types of cancer treatment. This includes surgery, chemotherapy, and radiation therapy. Once again, cancer is caused by certain changes to genes, the basic physical units of inheritance, as shown in this figure. In terms of cancer cases, here are the reported new cancer cases for these 4 countries, whereby in Malaysia, there were a reported of more than 37,000 new cancer cases last year. As I mentioned before, there are a few types of cancer treatment available, but they do have their own disadvantage. The disadvantage of current cancer treatment: for chemotherapy, it lacks of drugs localization, meaning that the drugs is very difficult to be targeted to the cancer cell site. For radiotherapy, it has large tendency of killing normal cells. For surgery, fatally damage healthy tissue, so not removing just cancer cells but also healthy tissue as well. So that's why I would like to describe in more detail about my invention or my product description. FICUS deltoidea or COTEK is a local plant that has been used in traditional medicine for a very long time due to its various health benefits. On the right hand side is a photo of FICUS deltoidea or COTEK leaves. Inside the leaves, there is a chemical compound known as catechin. Catechin, a flavonoid compound found in FICUS deltoidea COTEK leaves extract. Catechin exhibited several anti-cancer activities by eliminating the carcinogens or the cancer-causing agents and inhibiting the growth of cancer cells, meaning that it brings death to the cancer cells. Okay, before I go any further into hydroxyapatite, I would like to talk on nanoparticles. So what's so special about nanoparticles? Nanoparticles have large surface area, which is excellent attachment of pharmaceutical agent, drugs or genes on the surface of nanoparticles. Then it has the ability to penetrate cell and tissue, which improve treatment efficacy. This is due to its small size. And lastly, protection of pharmaceutical agent, avoiding reticuloendothelial system or REST clearance, meaning that the nanoparticles are very difficult to be eliminated by the human immune system. Okay, hydroxyapatite is a biomineral consisting of calcium and phosphate. So the advantages of hydroxyapatite for drug delivery is that it improves colloidal stability, avoid aggregation of nanoparticles, improve biocompatibility, meaning that it is safe for human uptake. In fact, human bone is composed of hydroxyapatite. And lastly, which is very important, provide protection of catechin, which promote protection of catechin from lysosome within the cells. So the cells will not destroy the catechin, which is the anti-cancer agent. Okay, so nanoparticle in cancer treatment could be explained the mechanism by this diagram. Firstly, nanoparticle carries the pharmaceutical agent, drug or gene. Secondly, nanoparticle will attach to the unhealthy cell. Thirdly, the nanoparticle is ingested inside the cell by endocytosis mechanism or the cellular uptake. And lastly, the nanoparticle breaks up and the pharmaceutical agent is released inside the cell. The pharmaceutical agent cause the abnormal cell to perform apoptosis, the cell death. So nanoparticle sizes between 50 and 80 nanometer are a requisite for optimal endocytosis and intracellular localization. So the nanoparticle size is very important for cellular uptake. Okay, in terms of novelty, okay, this is the first reported of encapsulation of FICUS deltoidea or mastrotate leaves extract with hydroxyapatite nanoparticles for potential cancer treatment application. This is also my PhD student research project, which she just graduated recently, and we also do have an intellectual property registration, which is patent filing, with the patent application number of PI 2014-702-579, with the title of a method for producing hydroxyapatite nanoparticles, whereby I have developed a method for producing hydroxyapatite as small as possible. You can find more details on this in Appendix 1. So now moving on to results. Here are TM images of the encapsulated catechin hydroxyapatite nanoparticles, which shows the primary particle size is roughly about 50 nanometer, which is very good for the cellular uptake. Here is the photograph of in vitro testing of the drug delivery in the lab, okay, on human breast cancer cell. If you see here, there are 4 separate photographs. A showing negative control, meaning that there is non-treatment carried out on that particular breast cancer cells. B, we're using a commercialized anti-cancer drug known as tamoxifen. C is just using pure catechin without hydroxyapatite, and D is the encapsulated catechin with hydroxyapatite. Photograph D shows that by introducing encapsulated catechin with hydroxyapatite, it kills most of the breast cancer cells, okay, as compared to other photographs. So it's a very efficient carrier of catechin to kill the breast cancer cells. Not only that, we did in vitro studies, but also we did animal testing as well. After introducing the encapsulated catechin and hydroxyapatite into live mice, after 3 days, we operated them and then evaluated the organs and the healthy cell. It shows that there were no abnormal growth of organ and also the healthy cell, meaning that there were no toxicity effects of the encapsulated catechin hydroxyapatite on the mice organ and healthy cells. So it is very safe for drug delivery and future anti-cancer application. In terms of commercial potential, feel more specifically in drug delivery application, this inorganic biomaterial complex has the potential for treating various types of cancer. Okay, in terms of publications, published in 3 very good journals and in 1 book chapter. For journals, number 1 is Power Technology, which is a Q1 journal with impact factor of 4.142. Secondly, in a Q2 journal with impact factor of 3.408, and lastly in a Q3 journal with impact factor of 1.365. All of these articles can be found in Appendix 2, 3, and 4. Okay, lastly, in terms of collaboration with USM for the in vitro testing and also animal testing, because we don't have that facility here in UNIMAP. Also, we had collaboration with Clinical Research Center at Hospital Tunku Fauzia on the aspect of drug delivery and cancer treatment. Okay, so that's all my presentation. Thank you to my honorable judges. I hope you will be having a very good day and be safe in this pandemic situation. Thank you once again.